Geneticists in our face and in the face of those geneticists who biologists can also interact with us, we can transmit genetic information and not only transmit, but and manage the biosystem, and practical application here is this most fundamental phenomenon, the discovery of how anything, name it, is so that we can manage biosystems, including the human body, animals, plants, viruses, and so on, and so on. The simplest thing you can do is kill pathogenic bacteria viruses. For example, the flu virus, they still can't fight it, nothing to do. As he lived, so to say, he lives in the same way. He kills people. I'm not talking about the HIV virus, AIDS, the so-called acquired immunodeficiency syndrome, then Ebola viruses, and so on, such viruses very much.
Вирус Эбола и так далее. Таких вирусов очень много. Патогенные бактерии, например, палочка Коха, туберкулез. До сих пор синтезируют уже там...
Pathogenic bacteria, for example, Koch's bacillus, tuberculosis, dozens and dozens of antibiotics are still being synthesized, and tuberculosis is not curable, practically. We set up model experiments based on the results of the translation of genetic information, working genetic information at a distance. We set up model experiments where we demonstrated what is possible, selectively remove unnecessary organisms, for example, viruses, bacteria. Here with bacteria, for example, we had this. Several mothers turned to me, whose children were infected with pathogenic bacteria, against which antibiotics did not work.
Поражены патогенными бактериями, против которых не работали антибиотики.
This is a problem now, a problem in medicine, because sustainability antibiotic resistance is growing because they are being fed agricultural animals tons of antibiotics, to grow faster, the meat mass set went faster, and so on, and when antibiotics are applied massively, then bacteria, they find very clever ways to get rid of these antibiotics, destroy them in your body. Such a very complex biochemical work is being implemented, thus bacteria, and they become resistant, and that's it, that is, it's a huge problem. So, what to do? Back in Toronto, when we were working, we showed that it is possible a sign of genetic sensitivity to antibiotics to transmit resistant, that is, bacteria that are resistant to antibiotics, to return sensitivity to antibiotics, not to synthesize all new antibiotics, but just to return sensitivity. It's in our hands, so to speak, and so on. In the simplest model experiment, when we took very well a plant studied in the genetic sense that it is called Arabidopsis thaliana or Resicovidca thalia, so also called, so we just killed this plant at a distance of 7 kilometers, causing him mutagenesis, a special mutagenesis that it is caused, so to speak, by what you give as source of disinformation, not information, disinformation, DNA molecules that are damaged. If you read information from them, and these molecules we extract DNA from this plant, these DNA molecules damaged, that is, they contain misinformation, we are transferring it to all 4 sides, selectively this information reaches these plants and they die at last. So, it is possible to fight weeds, for example, pests agriculture, which are killed by poisons, and these poisons and we are being killed. And we are environmentally friendly, you can gently influence for any unfavorable organism, we can program so-called stem cells. These are the stem cells from which you can get any organ or any tissue in the body to grow new kidneys, new pancreas, how we did it in Toronto, new vessels, new eyes, and so on. That is, you can replace the old ones with new ones and thereby solve the problem, the inhibition of aging, rejuvenation and so on, what humanity has been striving for thousands of years, but the result is still zero. So, it turns out, it is now possible with the help of our here are such quantum technologies, to program stem cells, that is, make them turn into those organs and the tissues that the patient needs. Well, for example, there are a lot of spinal patients, that is, the man suffered spinal cord injury. Modern medicine is not able to treat such patients to cure, they slowly die, they are motionless, sitting in a chair or lying down and nothing can be done about it. What should I do? It is necessary to regenerate the spinal cord. This task is beyond the power of modern medicine yet, but now, thanks to our research, this task solved. That is, we take information from certain bodies and tissues of abortive material that is simply thrown away, small embryos measuring 3 to 5 centimeters, which, so to speak, for some reason they could not develop like that in a normal form, because mom is sick, so to speak, and the embryos themselves are healthy. Here is such abortive material we take and, so to speak, we read information from the brain, from the kidneys, from the liver, from the pancreas, from the spinal cord, and so on, and so on, and so on. And we write it down, just take it and write it down. What is good about technology? It is, so to speak, purely externally it looks very simple. Although quantum physics is behind it, I'm not talking about that, this is a special conversation. That is, those quantum models, physico-mathematical models that physicists and mathematicians and linguists, who work with us, they have prepared all this and we published, that is, we have a solid theoretical base. So, such information can be considered, recorded on a flash drive, roughly speaking, and then grow stem cells, the cells of the patient himself, well, for example, spinal a patient who has been lying there for 10 to 15 years, fat is taken tissue, a small piece of fatty tissue from him, cleaned surgically, the so-called mesenchymal stem cells, it's very convenient. And, therefore, with the help of our equipment, secondary laser radiation, which we have already talked about, is a laser, which models quantum processes in chromosomes, we enter the information read from the organs of the tissue, from the kidneys, from the liver, from the spinal cord, from cells, from the neural spinal cord and so on, we inject into these stem cells, thereby we program them. Then we grow it in sufficient quantities and introduce it simply, intravenously to sick paralyzed people. And they begin to move with us. Even before they got on their feet and went to us, more it is necessary to go through certain stages, but some have already, they can already move on all fours, before that, they were absolutely motionless. This is the benefit, the practical benefit that we extract from those developments, experimental theorists, which we have. We are working on precedents, because we do not yet have there are many clinics and so on, that is, we demonstrate individual cases, that this is fundamentally possible. For example, a typo of the eye, right. Blind woman, right eye blind. What to do? We program her stem cells, and every organ, every the tissue has its own stem cells, including among these are mesenchymal stem cells that they are suitable for all cases, and we cause regeneration eye prints. That is, a person becomes sighted. Or other eye pathologies, we already have several of these there have been, and still are, cases when we have regained sight. The professor is like this, I won't mention his last name, very a famous scientist, blind, sees nothing. They made him such an individual program, he began to see. Or a child, a year and a half baby, he has a viral infection. The optic nerve is affected, he is blind. We give him a specific program. At the same time, it is very convenient to use this program. You have to listen to it, it's a sound. It's just a sound. But, according to the theory, there is work, here in particular Lucignano worked, which showed that this is a torsion bar or spinal information, it can be recorded in sound is the carrier of it. And here is this baby, who for a year and a half I listened to a program written for him for a month, he sees. Recently, Just yesterday, they brought me, showed me this baby, yes, he sees. It's not quite good yet, but it sees. You know, this is what medicine dreamed of. No one has ever done this. No one grew teeth. We have a precedent for the regeneration of teeth. Moreover, the person who worked with us, according to our technology, in fact, introduced information in the bud teeth. Removed from the follicle, from the rudimentary tooth, introduced in the table cages. Of a person, injected the dog in the jaw, at whose fangs were removed, 2 fangs were removed, and our information was entered there from the programmed table cages, and grew up in the dog, safely, 2 molars instead of canines. You know, this is nonsense for a medic, for a dentist, so to say, for an optometrist. What I'm saying is complete nonsense. It's the same for a virologist. And this is an opportunity, so to speak, only the first, so to speak, the implementations that we have made. That's what the brilliant prediction that I made means. Garyaev in the 1920s and up to the 1940s he wrote about it, working in Germany, and his, so to speak, from Russia and the Soviet Union was expelled. And where did he say that chromosomes have wave or field equivalent? That's what we did, and what I did means. Luc Montaigne, transmit genetic information at a distance. And the most important thing is to make this information sound. I got to geneticists, I got to biologists, I got to doctors, to the farmers, who, so to speak, score tons land with poisons to remove weeds. Thus, we ourselves are poisoned. All this can be avoided by using technology. Information, wave new genetics.